We will now discuss the retroviral life cycle in a little bit more detail. So we know that retroviruses are envelope viruses which have viral glycoproteins incorporated into the viral membranes. And these viral glycoproteins bind to specific receptor. Each retrovirus has very specific receptors. For example, HIV uses CD4 with either CCR5 or CXCR4 to enter. MLV uses amino acid transporter. Ape leukemia virus uses phosphate transporter, and so on. Once a surface glycoprotein binds to receptors, this triggers conformational changes which open fusion protein, which inserts into the target cell membrane and trigger fusion of viral membrane and cellular membrane. A viral nucleocapsid is released into the target cytoplasm and travels along the microtubule towards the nucleus. During this time, viral RNA is retrotranscribed into viral DNA. Once it reaches the nucleus, the viral DNA is transported into the nucleus. This can happen either when the virus is allowed to reach chromosome and integrate because cell proliferate. You recall when cell undergoes mitosis, nuclear membrane is dissolved, so virus have free access to the cell chromosomes, or some viruses have nuclear localization signals. Their matrix and integrates as NLS so they can mediate active transport to nucleus. Once the virus is reaching the chromosome and integrated, it forms so-called provirus. And provirus can stay silent in the target genome, or it can be transcribed and trigger synthesis of mRNA and translation of viral protein and synthesis of the virus. Now, let's talk a little bit more about retrovirals reverse transcriptase. It is one enzyme which has three different activities. A retroviral reverse transcriptase function as RNA-dependent DNA polymerase, which synthesizes DNA on the template of RNA. It also functions as a DNA-dependent DNA polymerase, which synthesizes DNA on the template of DNA, and it also has ribonuclease H activity, which degrades RNA in the hybrid of RNA-DNA. So all these three activities are mediated by reverse transcriptase by one enzyme. Now, when DNA is synthesized, it is part of so-called pre-integration complexes, which are targeted to nucleus, and within nucleus integrase mediate integration of DNA into target chromosome and creates proviral DNA. Now, reverse transcriptase lack proofreading capabilities, which results in many mutations. So it generates quasi-species. DNA is altered sequencing and with altered characteristics. So reverse transcription is very tightly regulated stepwise processes which undergoes two template switches. You remember viral RNA is plus sense, so it goes from 5 to 3 prime, and on both 5 and 3 prime it has these repeats, and on 5 prime it has PBS primer binding site. You recall we said that virus also packages cellular tRNA into virions. And this tRNA serves as a primer which binds to primary binding site and triggers synthesis of the short DNA. You recall, since the viral RNA goes 5 to 3 primes, this has to go opposite direction. So it synthesizes very short DNA, which is called strong stop DNA. This creates a DNA-RNA hybrid, so the RNase H activity of RT degrades this RNA up to PBS. So now the short DNA must switch template, and it switches to 3' end. It can be 3' end of the same RNA or second RNA. You recall the retroviruses package 2 RNA. Now it can anneal here because there are these repeats which are complementary. And from there, it synthesizes DNA up to PBS primers and create another DNA RNA hybrid. So the RNA, original RNA, is degraded except of this short PPT polypurine tract. This one is protected from degradation and further serves sorry, further serves as a primer for synthesis of the complementary DNA. You recall, so it goes through U3, RU5, and PBS. So now the short plus strand strong stop DNA needs to switch template again, but it has this PBS region which would anneal to 5' and PBS and synthesize complementary DNA. This process yields this 2 LTR long terminal repeats on 5' and 3', and both have U3 RU5 sequences. 5' LTR serves as a very strong promoter for viral transcription. Now, once the DNA is synthesized and targeted to nucleus, integrase mediates integration into the target genome, and it triggers cleavage of target DNA and insertion of the viral DNA, which becomes provirus. Proviral DNA integrated in the target DNA is random, meaning integration is random. There is no specific site or specific chromosome to which a retroviral DNA would integrate. However, it usually integrates in the area which is transcriptionally active, which makes sense because when genes are transcribed, the DNA is open, the condensation is released, and the DNA is accessible to the proteins and transcription factors as well as the viral DNA. So it is random, but mostly found in the area of active gene transcription. Once the provirus is integrated, transcription can start, and it starts from LTR. Usually there is exclusion of 3' LTR, and transcription start from 5' LTR, which serve as a promoter. And upstream of U3R junction, there is TATA box, which you recall is very important for general transcription machinery to anneal. Transcription generates mRNA. GAG-POL, you recall, mRNAs are not unspliced, but envelope mRNA is spliced, okay? And they are also polyadenylated, and they have capping. Now, how the polyprotein can be synthesized? When the mRNA, which contains GAG-POL, is synthesized, so the synthesis of polyprotein is enabled by alternative reading frames. You can see here there is one reading frame. Focus on these heptamer sequences, which are labeled in red. This is frame 1, which start to read from the second A here, and this is frame 2, which start from the first A. So those are so-called alternative reading frame, which change from reading frame 1 ends with the stop codon. However, when the reading frame 2 is shifted to, this actually changes the stop codon, and now the reading through can happen. So ribosomes can actually read through. So polyproteins can be synthesized from these mRNAs because of the alternative reading frames. Once viral proteins are synthesized, the envelope proteins, which contains envelope proteins, glycoproteins, surface glycoproteins, and transmembrane, they are targeted to the cell membrane where they are inserted, and GAC-pol are attracted to it because the matrix is myristylated and bring whole GAC-pols into the area where the glycoproteins, surface glycoproteins are inserted in membrane. Nucleocapsid portion has packaging signals, so it attracts viral RNAs and virus assembles on the membrane. It exits cells by budding and forms immature virions. Only when immature virions is released, the viral proteases are activated and cleave polyproteins into functional final proteins, which generate mature virus. Now, let's go a little bit to discuss oncoretroviruses, those which have acute transforming capability. These viruses captured some proto-oncogene from cells when they were synthesized from integrated provirus and mutated and turned that proto-oncogene into oncogene. You know that proto-oncogene are cellular genes which have normal function during cellular replication. However, if they are mutated and acquire continuous active state, they become oncogene. For example, Rao sarcoma virus picked up SARC kinase, which is mutated, and when it in fact targets cells, it triggers uncontrolled proliferation. Similarly, avian erythroblastomatosis virus pick up another tyrosine kinase receptor ERBB, and avian myelocystomatosis virus pick up MYC. MYC is very interesting transcription factor because it regulates over 15% of our gene transcriptions. However, what is very important from the point of the transformation is that it also regulates synthesis of the cyclins. And we know cyclin activates CDK. CDK enables promotion of the cycling. So MYC brought to the cells functions as oncogene. Now, some retroviruses do not carry oncogenes, and they still can trigger oncogenesis. This may happen when a retrovirus is integrated upstream of some proto-oncogene. And in this case, if the 5' promoter LTR is inactivated, 3' LTR functions as promoter and therefore transcribes the downstream sequences, which as a proto-oncogene would push cell to proliferate. This is a very interesting feature, and it's why there is huge hesitation to use retroviruses as a viral vector for delivering of gene or for vaccine, because they integrate randomly and we cannot control whether they will be integrated upstream of some proto-oncogenes. Now, at the end, let's talk a little bit about endogenous retroviruses and retrotransposons. Approximately 8% of human genomes is composed of retroviral DNA, so-called endogenous retroviruses. Upon the insertion of the viral genes, they lost some capabilities, they lost some sequences, so they cannot synthesize functional infectious viruses, but they can still, upon certain circumstances, activate synthesis of particular genes. Even more intriguing are retrotransposons. They form over 40% of human genomes, and they are formed or composed of retrovirus-like elements. So they obviously contributed to the evolution of our genome. So transposons, I recall from the genetic too, that those are what we call jumping genes, which can jump from one location to another, and retrotransposon are part of the transposable elements or jumping genes.